So if we consider the origin of eukaryotic cells, we must account for the major differences between the eukaryotic cell and the prokaryotic cell. And one of those key differences, as we've already discussed, is the membrane-enclosed nucleus, which partitions the genetic material in eukaryotes from the rest of the cell. And that nuclear envelope or nuclear membrane does not exist in prokaryotes. Rather, the DNA in prokaryotes is in a nucleoid. So some recent molecular evidence points to the origin of the nuclear membrane in a modern eukaryote, the double membrane system of a modern eukaryote, points to the origin of that double membrane enclosing the nucleus of the eukaryote. It points to an origin that involves an invagination and curving of the plasma membrane in an early endomembrane system in an early eukaryote. And then this would achieve a greater surface area for diffusion of substances in and out of the cell as eukaryotic cells grew larger than their prokaryotic counterparts. And in order to do this, there must be proteins that curve the membrane that allow for these types of membrane canal systems to exist. And if you look at a modern eukaryote, there are proteins that curve membranes that exist at the ends of endoplasmic reticula canals. And also, as we'll see, there are vesicles that bud off from the various endomembrane components and exist as vesicles that can carry molecules to various parts of the cell or can fuse with the plasma membrane and dump contents to be exported out into the extracellular space. And if you look at those proteins that exist at these positions, especially the ones that are in what we call coated vesicles, which are trafficking vesicles inside the cell, and look at the proteins in those coated vesicles and look at their structural architecture, the types of alpha helices and beta sheets and the motifs that those exist in, you find that some of the proteins in the nuclear pore complexes, which are macromolecular assemblages that exist at the nuclear pore, you find structural homology between nuclear pore complex proteins, some of them, about 7 of them, and some of the proteins that exist around coated vesicles that curve the membrane. And so the idea is that the nuclear envelope evolved from invaginations of the plasma membrane as part of a canal system to increase surface area to volume ratios. And you can witness the evolutionary origin of the proteins at the nuclear pore complex by looking at their structural homology to proteins that exist elsewhere in the eukaryotic cell today that are involved in curving membranes. So we think the nuclear envelope and the nucleus evolved in an early eukaryote by being surrounded by these invaginations of the plasma membrane and finally becoming secured in a modern eukaryote in an enclosed double membrane system. Note that this does explain why there is a double membrane system, not a single lipid bilayer like there is at the plasma membrane, because the double membrane system would arise from this type of invagination. So that's kind of an interesting feature of an interesting feature of the evolution of the eukaryotic cell, that it can be inferred from molecular homologies that exist between proteins in the eukaryotic cell today. Now let's leave the evolution of the eukaryotic cell or further treatment of the modern eukaryotic cell until a little bit later, and now let's visit some other components, and we'll get back to evolution of the modern eukaryotic cell shortly. So let's now consider some other components of eukaryotic cells, like ribosomes. We know that ribosomes are the site of protein synthesis in the cell. They have 2 components: a ribosomal RNA that is transcribed from genes in the nucleus and forms structural and also functional components of the ribosome, as well as proteins. So we refer to ribosomes as a ribonucleoprotein complex. And they are large. Ribosomes are very large. They're found within the cytoplasm, and attached to internal membranes as well, as we'll see. So here's a blow-up of our schematic of the cell that we looked at earlier. And here we see ribosomes both attached to the membrane of the rough endoplasmic reticulum, as well as free ribosomes in the cell. And if we look at the structure of a ribosome, we see that it's composed of a large subunit and a small subunit. Excuse me. And these subunits, again, are complexes between RNA and protein. And we will look at the structure of a ribosome in a little bit more detail right now. So this is a ribosome from an archaea. We know that all domains of life, the archaea, the prokaryotes, and the eukaryotes, have ribosomes, the protein manufacturing factories of the cell. And this archaeal ribosome has a large subunit on top, and we're rendering the small subunit below. We refer to the size of large macromolecular assemblages like the ribosome, and we refer to the size of those in Svedberg units. So we have S units. That is a Svedberg, after a Swedish biologist who figured out how to use centrifugation to separate large macromolecules. And we see that the large subunit here, not surprisingly, has more Svedberg units. It sediments in centrifugation at a higher mass than the lower subunit. So the top subunit here is called a 70S subunit. And so this is the 70S subunit up here. And the small subunit down here is the 30S subunit. So we have a 30S subunit and a 70S subunit. And that would be down here and up here. So let's look at those now in a little more detail. Here we're rendering the RNAs starting out in red and the proteins in tan. And we're now going to render the protein components and the RNA components some slightly different colors so that you can resolve them. So here we have, in this large subunit on top here, we have a large RNA, a 23S or 23 Svedberg RNA shown in silver ribbon. And then there's a much smaller RNA here shown in magenta that is the 5S RNA. Whereas in the smaller subunit here, we have a 16S RNA shown in light blue ribbon. Now in both subunits, besides the RNAs that are present, you see proteins, and we're rendering those in a ribbon rendering so that you can see the alpha helices and beta sheets where they exist. You can see the secondary structure. So the proteins for the small subunit are in dark blue, and the proteins for the large subunit are in dark purple. Now if we focus in on the small 30S subunit down here and look at the 16S RNA that is present, is the component of that small subunit, we see that that RNA, that 16 Svedberg RNA, has three major domains. It's one RNA molecule, but it has three major domains. Actually, four major domains, sorry. And there are the proteins shown in spacefill. So now we're rendering the small subunit 16S RNA in light blue, and the proteins you'll see are located in the small subunit rather at the periphery. The core of the small subunit really is the 16S RNA. Let's look at those domains one more time. The 16S RNA of the small subunit has four major domains. This is the 5' domain, the central domain, the 3' major domain, and finally the yellow domain is the 3' minor domain. 5', 3' refers to the polarity of the RNA chain. And here are our proteins shown in spacefill, dark blue. Let's now move to the large subunit and look at the 23S RNA of the large subunit. And here we're seeing that there are 6 major domains, but they're not nearly as separated as the domains in the small subunit. They're rather much more intertwined. So we're just rendering the 6 domains in different colors here. And in addition to the 6 domains of the 23S RNA of the large subunit, there is also the 5S RNA here, the small RNA of the large subunit. And as for the small subunit, you can see that the proteins shown in dark purple are really primarily at the periphery of the large subunit. The core of the large subunit, the heart of the subunit, is really composed of the RNA components of the large subunit. So the ribosome is a ribonucleoprotein complex that has major RNAs, two RNAs in the large subunit, one ribosomal RNA in the small subunit, and many proteins, over 30 proteins, 20 proteins in the small subunit, and 30 proteins in the large subunit. Now you'll remember we pointed out that the RNAs of the small subunit, the RNA of the small subunit, the 16S RNA, has 4 major domains that are quite separate, whereas the domains of the 23S RNA in the large subunit were not quite as separated as the domains of the small subunit RNA. And that implies that the small subunit undergoes extensive conformational changes as the ribosome moves down a messenger RNA, translating that messenger RNA into protein. And we will revisit this in greater detail later, but it's good to introduce the structure of the ribosome at this point even before we get to the mechanisms of protein synthesis that this amazing macromolecular machine accomplishes. So let's move on now from the ribosome to the endomembrane system. And the endomembrane system is a series of membranes or canals throughout the cytoplasm. And these really compartmentalize the cell into places, into locations where different cellular functions are achieved. The first component of the endomembrane system is the endoplasmic reticulum. And we're going to also consider the Golgi apparatus, and finally lysosomes as part of the endomembrane system. So if we look at the endoplasmic reticulum, there is the rough endoplasmic reticulum, the RER, and these are membranes that create a kind of a canal system, a series of channels throughout the cytoplasm, and the rough endoplasmic reticulum has ribosomes attached to the membrane. And those ribosomes, as they manufacture proteins, shunt the proteins into the lumen, or the inside of the rough endoplasmic reticulum. And there, those proteins can be encapsulated in membrane vesicles that bud off from the rough ER and are transported to other components of the endomembrane system, the Golgi, and then from there they can be modified and exported to the cell surface, where they can be shunted into the extracellular space, or alternatively can embed in membrane and become part of the plasma membrane. We'll go over that in more detail a little bit later, but it is the attachment of ribosomes to the membrane of the endoplasmic reticulum that renders it the rough endoplasmic reticulum. It's called rough because that's the way it appears in electron micrographs. So again, the synthesis of proteins on ribosomes, those proteins that are going to be secreted or sent to lysosomes or the plasma membrane, are made on the ribosomes associated with the rough ER. Now in the next part, we'll pick up with the smooth endoplasmic reticulum and then move on to other cellular structures in the eukaryotic cell.